Hi, my name is Jade Davis and I'm a molecular genetics student. I completed my Pawsey summer internship at the Minderoo Foundation's Oceanomics Centre, helping to optimise their code for cetonics. So one interest of the Minderoo Foundation is in protecting marine biodiversity, which is at risk from various factors such as overfishing and climate change. One way in which this is monitored is through a process called environmental or eDNA analysis, during which researchers will collect seawater samples, extract the DNA in it, and then compare this DNA found to a reference genome database to identify which fish species are in the area. For this analysis, they require high-quality reference genomes of the marine animals so they can match up this DNA in the samples to the reference genome database. However, currently only around 1% of the known marine fish species have had their genome sequenced. And therefore, at Oceanomics, researchers are working to construct reference genomes for these species which haven't had those genomes sequenced. One of the programs they use to create these reference genomes is called Diploidocus, which specifically analyzes diploid genomes. However, this does not run very efficiently on cetonics because there's just one code that runs through a variety of different steps sequentially. So this means that the code will both run very slowly because you can't parallelize the execution of different steps, even if they aren't dependent on each other. And it also means that the code inefficiently will use computer resources because you must request a certain amount of memory, which won't necessarily be needed by every single step in the process, and also a certain amount of CPU, which again might not be efficiently used by all steps. So to optimize this code for cetonics, I use Nextflow to create a workflow which separates the different processes out so that steps which don't depend on each other can be run in parallel. I then benchmarked each process to find the ideal CPU and memory allocation that would speed the process up the most without drastically increasing the service unit cost. I used the findings from this benchmarking to create easy-to-use workflows for genomes of differing sizes. So because the cetonics usage cost is based on the highest percentage of memory or CPU usage, I found that the best way to do this benchmarking was by working out how much memory a process needed to run, and then working out how the program ran at an equal cost of CPU, since most processes required a significant amount of memory. Overall, these changes reduced the code's overall execution time by an average of 30% and decreased the service unit cost by an average of 40%. So this code optimization will speed up this step in reference genome curation for oceanomics users, therefore streamlining the overall process of creating marine genomes for biodiversity monitoring. It also opens up the use of Diploidocus to researchers using high-performance computing to construct diploid genome assemblies in other fields of genomics, such as agriculture or conservation, since they will be able to use it more easily on high-performance computing now. Overall, this was a very valuable and interesting process that I'm so glad I got to be a part of, and thank you for listening to my presentation.